Start entering your attendance in the chat box. Can you see the screen? One of you unmute yourself.
Yes, ma'am.
Okay, let
Yes,
us begin now. Till yesterday, we discussed different organelles in prokaryotes as well as eukaryotes. We discussed 4 different organelles in the last lecture. Now we'll continue in the same. That is the next and the most important organelle which is present in the cell, which is considered to be the mastermind of the cell, and that is called as nucleus. So nucleus is a cell organelle which was described by Robert Brown in early as 1831. So in case of this structure in detail, the structure as well as functions will be studied now. The nucleus was stained by the basic dyes and given the name as chromatin by Fleming. Now you can see this structure where you can see these rounded structures. These are the proteins and wrapped by the DNA. So chromatin is nothing but which consists of nucleoprotein. That means nucleic acids and proteins. Now which type of nucleic acids we'll discuss. The term chromatin was coined by Fleming, and this consists of DNA, that means nucleic acids and proteins. Now this is the ultra structure of nucleus which is there in your textbook also. We will learn this. Now in case of plant cell as well as animal cell, you can see such type of structure. This is what the entire structure called as nucleus. Now what are the parts seen in the nucleus? Now you can see this surrounding part, that is the double membraned structure. That is the 2 membranes you can see here, that those membranes, that is outer and inner, both the membranes together, which bounds entire material inside it, that is the boundary of the material inside this, that is called as nuclear membrane. So nuclear membrane, this boundary of the nucleus is called as nuclear membrane, which is divided into outer nuclear membrane and inner nuclear membrane. So 2 parallel lines which form or make the space in between these 2, that is called as perinuclear space. Peri is what? Surrounding. So what it is surrounding? That is the nucleus. So the space which is present in between these 2 layers, that is what called as perinuclear space. So this is the first part of the nucleus, outermost layer which covers, which surrounds, which bounds the nucleus is called as nuclear membrane, which is double layered structure having the space or the gap in between it, that is called as perinuclear space. Inside it, you can see this thread-like structure, long, elongated thread-like structures, and that is nothing but the what this is called as chromatin material. What this chromatin material consists of? This chromatin material, as I said, the thread-like structures that consist of proteins, that is histone type of proteins, as well as nucleic acids. So that is nothing but the nucleic acids and proteins. Not only that, in the inside this nuclear membrane, semi-solid or you can say aqueous material is present, and that is called as karyolymph, or this is also called as nucleoplasm. The plasma which is present inside the nucleus, that is called as nucleoplasm, and the same is called as karyolymph. Lymph is semi-solid or aqueous substance, and karyon means nucleus. So whatever that material present, aqueous solution or this aqueous solution present, that is called as lymph, inside the karyon, that means nucleus. So this is called as karyolymph. So first is what? Membrane, that is nuclear membrane. Second is what? Inside the nuclear membrane, whatever the liquids, aqueous solution or semi-solid or aqueous jelly like, or you can say aqueous solution is present, that is called as karyolymph or nucleoplasm. Nucleoplasm and karyolymph are one and the same. Okay, karyo means nucleus, lymph is aqueous solution, that means that which is present inside the karyon, that means nucleus. Inside this nucleoplasm or in the nucleoplasm or the karyolymph, different type of substances like nucleic acids, proteins, some enzymes, ribosomes, such type of structures are present, and these are embedded in the nucleoplasm. These are seen in the nucleus. But the main part which is called as chromatin, which looks like, which gives the appearance of thread-like structure. So that is called as chromatin material or chromatin threads. In undividing nucleus, it looks like that. So that is nothing but the histone type of proteins plus nucleic acids like DNA, RNA. So DNA, RNA, then proteins like basic proteins called histone proteins and non-histone proteins are present in the eukaryotic nucleus. Then one more thing you can see here, and that is here you can see the spherical structure, rounded structure, that is called as nucleolus. That nucleolus, it shows it is not bounded by any membrane. You can't see any membrane around it. But it gives an appearance of small, you can see the crystal-like structures. These are nothing but the ribosomal RNAs and ribosomal proteins. So these, it contains the material required for the protein synthesis. That nucleolus is considered to be important in biogenesis. That is, the synthesis of ribosomal biogenesis is nothing but that material is required for the synthesis of ribosomes. That means ribosomal proteins and ribosomal RNA is present in the nucleolus. So what did you see in the structure now, in the ultrastructure of nucleus? That is first nuclear membrane, which bounds, surrounds the nucleus, which is made up of two layers, outer and inner membrane. The space is present in between these two membranes, that is called as perinuclear space. Then this outer or inner, that perinuclear space, that, or you can say this membrane, nuclear membrane, which contains the aqueous solution, that is called as karyolymph or nucleoplasm, which contains chromatin material, that is in the form of nucleic acids and proteins, the thread-like structures. Then next is nucleolus. Sometimes single nucleolus is present, or sometimes many, two, three, or many nucleoli are also present in the nucleolus. Depending on the activity of the cell and the requirement of the proteins, the nucleoli number varies. In general, a single or sometimes two nucleoli are present. Nucleolus is singular, nucleoli is a plural. So generally one nucleolus is present, but in some cells, as per the requirement, many, two, three, or many nucleoli are also present. Then one more thing you need to remember, that chromatin material which is present, if the DNA, amount of DNA is more than the protein, then when it is stained, it gives, that stain gives dark color, that is darkly stained, and that is called as euchromatin. Try to understand. Euchromatin means when DNA concentration is more than proteins. The amount of DNA when is more than the amount of protein, it gives dark stain, it is darkly stained. Or that amount of DNA When it is more than the amount of protein, that is called as euchromatin, which is considered to be highly active. Whereas when the amount of DNA is less than the amount of protein, that means amount of protein, if it is more than the amount of DNA, then that is called as heterochromatin region. So these regions of chromosomes, they are stayed as per the amount of or the quantity of the DNA and the proteins in present in them. Okay, so in ultrastructure of nucleus, I will repeat again that first is nuclear membrane, which surrounds the nucleus, inside which whatever the active solution is present, that is called as karyolymph or nucleoplasm, which contains different substances like chromatin material, that is the DNA, RNA, proteins like histone or basic, or you can say non-histone type of proteins, some ribosomes. Then also it shows the presence of nucleolus. Single or many nucleoli are present, which is considered to be the place of synthesis type of ribosomes, that is ribosomal biogenesis, that contains rRNA, that is ribosomal RNA, and ribosomal proteins. Then one more thing you can see here, that there is a gap between the two nuclear membranes. That means not the two membranes inside, that is perinuclear space, but you can see some pores or openings in between that entire nuclear membrane, and these are called as nuclear pores. These all are called as nuclear pores. These gaps where you can see in between the membranes, these gaps are called as nuclear pores. And what is the role of nuclear pores here? A ball-like structure or bearing ball, how it is working, it is present over there. That is, that controls the entry and exit of the material inside the nucleus or from outside, or from the nucleus outside the nucleus. So entry and exit of the substances or the material from the nucleus, either inside or outside the nucleus, that is controlled by nuclear pore. So these all nuclear pores, they are playing very important role in the uh, cell, where the entry and exit of the substances like DNA, RNA, ribosomes, proteins, etc., or ions, this takes place through these nuclear pores. Nuclear pores are guarded, or there a bearing ball like structure is present, which is not seen here in the this diagram, but it is present, which works or which controls the entry and exit of the substance from the nucleus. So remember all these parts. So this is the ultrastructure of nucleus. And you know that as it contains the genetic material in the form of DNA, that is meant for heredity and variations. So nucleus is considered to be the master copy or mastermind of the cell. Now we'll see what is perinuclear space. As I said, explained in the previous diagram, that it consists of two parallel membranes with a space that is generally 10 to 50 nanometer in between, and that is called as perinuclear space. You can see nuclear pores very clearly in this picture, in this diagram. This is the entire nucleus, and this is the nucleolus, and these the small openings you can see, these are called as nuclear pores. So nuclear envelope is interrupted by minute pores formed by the fusion of its two membranes, and these nuclear pores are the passage through which movement of RNA protein molecules take place in both directions between the nucleus and the cytoplasm. That means inside the nucleus, nucleoplasm is present. Outside the nucleus, cytoplasm is present. In the cytoplasm itself, different organelles are present, which we are discussing. Larger and more numerous nucleoli are present in cells actively carrying out protein synthesis. As I told you, as these nucleoli, they are made up of, or they contain ribosomal proteins and ribosomal RNA, which are meant or which are responsible for protein synthesis. So as per the requirement, if the cell is actively participating in protein synthesis, then the number of nucleoli gets increased. So as per the requirement of the cell, number of nucleoli varies. But during different stages of cell division, cells show structured chromosomes in the place of nucleus. Now, what is that? That point we will discuss in the some other chapter. We will be having that part also next to this. See, when you see this, this is the chromosome, and this structure varies from during the cell division. Now, in the previous diagram, whatever the structure you saw, that is thread-like chromatin network, right? But when the cell is in the division state, cell is in the dividing nature or dividing form, then it changes its shape. The chromosomes, they change their shape. And so what type of shapes are seen, that we will discuss in the next chapter. Now again, this is what chromatin material. It contains DNA, as I said, RNA, proteins, basic proteins, they are called as histones, and non-histone proteins. Remember, these are in points. Now, how this is seen? This is double-stranded nature of nucleic acid. So that's why deoxyribose nucleic acid, that is called as DNA. RNA, this is single-stranded ribosomal nucleic acid. This is a type of RNA, one of the type of RNAs, that is mRNA, and this is the structure of protein. And now in this structure you can see these are the histone molecules wrapped by DNA. So this is the structure of chromatin.
Just a minute, madam. Just interrupt you. Prem Vagole, your mic is on. Switch it off. Prem Vagole, switch off your microphone.
Okay. Now this is what we studied, the structure as well as functions of nucleus. I hope you have understood the structure in detail, and the function also. As I said, that the main function what the nucleus is containing, that is genetic information, which is very important role it is playing, nucleus, for heredity and variations. And one more thing, that is the site of synthesis of DNA, RNA, and ribosomes. As I explained just now, that nucleoli, they are responsible for it. Then also the next function of the nucleus is what? It plays important role in protein synthesis. Chromosome number being constant for a species, it is important in phylogenetic studies. Nucleus thus is the master cell organelle. Remember, now on page number 55, you can see that the last paragraph that shows the functions of nucleus. You can keep your textbook open, page number 55, left hand side column, last paragraph, below the ultrastructure of nucleus, whatever the last paragraph is there, that shows all the functions of nucleus. You can give the numbers to it. First number, that is the genetic, it contains genetic information, so playing important role in heredity and variations. Second, site for synthesis of DNA, RNA, and ribosomes. Then the next, you can see, give the number that is being constant for species. Now you know how the number is constant for species, that is human being. How many chromosomes are present in human being? Same way, each and every species having its number of chromosome constant for that particular species. Okay. Now, the different Remaining few organelles which are present that we will discuss, that is cytoskeleton. What is cytoskeleton now? Skeleton, the word itself suggests, that gives a framework. Frame. Skeleton is what? Frame. Frame of what is present in the cytoplasm of that particular cell, which is giving a frame. Frame means what? A particular structure, or it makes a particular form, right? So that particular network of filamentous structures which are proteinaceous in nature, which gives a particular form or shape or structure in the cytoplasm, is collectively referred to as cytoskeleton. Okay, so cytoskeleton, the word itself suggests. Skeleton means what? In case of human beings also, you know what it makes the skeleton. It is giving a form, it is giving a shape, it is giving a structure, a particular structure, and that is what? Skeleton, which is present in the cytoplasm of the cell. That is what? Network of filamentous proteinaceous structures present in the cytoplasm is collectively referred to as the cytoskeleton. Remember, now what are these eukaryotic cytoskeletons? First, microfilaments. Second, intermediate filaments. And the third is microtubules. It's there in your textbook also. You can underline. You can also underline as well as note it down. These filaments or these structures which are in the filamentous form, they are made up of specific type of proteins. So that's why they are called as proteinaceous structures, forming a specific forming a network with a specific form or shape or structure. Okay, so microfilaments, intermediate filaments, and microtubules, these are the three type of proteinaceous filaments. Microfilaments, they are made up of a specific type of protein called as actin. Actin is a protein. Then microtubules, they are made up of protein fibers or tubular proteins. They are called as tubulin. So tubulin is the protein. Microtubules are made up of that protein called as tubulin, giving the tubular structure, tubular form. And intermediate filaments also, they are made up of different types of protein subunits like keratins. So these are fibrous subunits. So these three types of filaments which are made up of specific type of proteins make a structure or frame. So that's why they are called as cytoskeleton. They are giving the skeleton or shape or structure to the cell. So what are the functions? It gives mechanical support, then maintains the cell shape. Because of these filaments only cell shape is maintained. Also it gives mechanical support or strength. Then with the help of filaments, cell can show motility. Then intracellular transport is also done within the cell. Different organelles within the organelles, that is within the cell, in between the organelles in the cell, there is the transportation of the substances that is done with the help of these filaments. Different types of cytoskeleton filaments. Then signal across the cell. While crossing the cell, some types of signals are important that are given with the help of these skeleton, cytoskeleton. And karyokinesis. Kinesis is division, carry on, that is nucleus. During the nuclear division also, they are playing very, very important role. So that what exactly role is that? During the type of cell division we will be discussing. Now the next important organelle, that is cilia and flagella. Cilium is singular, like that flagellum also singular. Cilia and flagella are hair-like outgrowth of the cell membrane. Cilia are small structures causing the movement of either the cell or the surrounding fluid. Now, in this picture, you can see paramecium with all small hair-like structures which the entire body it is surrounding. So, this is what they are called as cilia. So the combined together movement in coordination, the movement of the cells is in the coordination with each other. That is what the role of cilia. They are small. This is what in this structure you can see in the euglena and bacteria. These are long thread-like structures, either a single one or few, but the number is less in case of flagella as compared to cilia. Cilia are generally present all over the body. Flagella, either a single or few, either at one side or at opposite ends, or sometimes all over the body, but very rarely. So these are, that is the difference between cilia and flagella. Now when we study the internal structure of cilia and flagella, you can see this part. What are the cilia and flagella made up of? Three parts generally they are seen. That is, first is basal body, then the basal plate, and this is what the structure called as axoneme. The central part, that is the core, the entire core part, central part, that is called as axoneme, and that is covered by a sheath that is called as sheath. That is covering. Covering is what? Sheath. So this entire structure, the core of this flagella or cilia, that is called as axoneme, whereas this is the plasma membrane or the plate, basal plate, and that is the basal body. So these are the parts. Now internal structure, when we see very clearly, you can see these different parts. If you count here, these double ring like structures you can see here: 1 2 3 4 5 6 7 8 and 9. So these 9 double rings are present on the periphery, right? Whereas two single ring like structures are present at the center. Then one more thing you can see, that these spikes or these radiating structures which are called as spokes, they are present, which are connecting this central microtubule with the radial one. These are these radial double ring structures, they are called as dyname arms. That is a type of protein. They are made up of, they are called as arms. Whereas this is the central core. There, these are called as central microtubules. So outer are doublet microtubules and inner are single one. These are two. Then one more thing is there, that each central microtubule is connected with the peripheral or the radial doublet microtubule. Then each doublet peripheral microtubule also is connected to each other. Then one more thing you can see, that these two central microtubules are also connected to each other. So I will repeat again the parts of this cilia and flagella, ultrastructure, or in the structure of cilia and flagella, which is called as a 9+2 type of arrangement in case of eukaryotic cilia and flagella. What is 9? These are 9 in number, these double microtubules, doublet microtubules, which are present at the periphery. They are 9 in number, and 2 are present at the center. So this type of arrangement is called as 9+2 type of arrangement, specially seen in eukaryotic type of cilia and flagella. Now in the second structure, you can see here this diagram. The core of cilium and the flagellum is called as axoneme. Core, that is the central part, that is called as axoneme. These are nothing but the microtubules, and they are surrounded by such 9 doublet ring-like structures, right? Peripheral structures, radiating structures, which are connected to each other also, and which are connected to the central tubules also. So the center or the core of the flagellum or cilium, that is called as axoneme. This is the 2D, very simpler form or model or structure of cilium or flagellum. Cilia and flagella. So this is called as 9+2 array. Now you can see here, these are the doublet peripheral ring-like structures. These are the doublet microtubules. This is a single microtubule. This is another microtubule. Two are joined. So doublet form of microtubules, which are arranged at the peripheral region. and these are single microtubules present at the center. And if you see, these two are connected to each other by the microtubular bridges. These are called as bridges. Also, each doublet is connected to each other by the bridges. Then you can see again these, the entire, they also are connected, peripheral are connected to the central one by these spoke like structures, right? So this is the structure or ultrastructure of cilium or flagellum, that is called as 9 plus 2 array. So the axonium usually has 9 pairs of doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. This type of arrangement of axonal microtubules is referred as the 9 plus 2 array. The central tubules are connected by bridges. Here, these two tubules are connected by bridges. Here also you can see, and here also in the diagram you can see. Central tubules are enclosed by central sheath. That is the sheath, as I said, this core part, axoneme, is covered by a sheath, plasma membrane. This is sheath. So, both the cilium and flagellum emerge from centriole-like structure called basal body. This is the basal body, that is the base of cilium and flagellum, from where it arises with the core as axoneme and its sheath which surrounds it. Now the next part, that is called as centrosome and centrioles. Centrosomes are made up of protein called as tubulin. So these are all proteinaceous in nature, and that protein is tubulin type of protein. They are made up of 9 evenly placed peripheral fibrils of tubulin. Each of the peripheral fibril is a triplet. Now in this diagram very clearly you can see that this is the central core and these are the peripheral microtubules which are in combination, that is 3 in 1. You can see 1, 2, 3. That is called as triplet. So these are all peripheral. They are present at the peripheral region. So this is what called as peripheral fibril, that is in the triplet form. These fibrils together in 3 together are present. So that is what triplet. So these are peripheral fibrils and they are made up of tubulin. This is the structure of centriole. So these are all microtubules which are in triplet form and they are present at the periphery. In this diagram again it is clearly seen. In your textbook also the similar type of diagram is seen given. So this is the core hub, and then here also in this diagram you can see this part, central part hub, and this peripheral region microtubules in the triplet form. You can count also 1, 2, 3, 4, 5, 6, 7, 8 and 9. So these are 9 in number. That is the microtubules made up of tubulin in triplet form present in the peripheral region. What is centrosome? Soma means body, which is present at the central place. This is what which is present near the nucleus of the cell. In the eukaryotic cell, these centrosomes are present near the nucleus because they are playing very important role during the process of cell division. That cell division which shows the division of nucleus followed by division of cytoplasm. So their place is near the nucleus because they are playing very important role during the division of the nucleus followed by the division of cytoplasm. So the actual role you will come to know when we will be discussing the cell division part. But you just remember right now that centriole, centrosome, these are playing an important role during the process of cell division, like mitosis, meiosis, you will be studying in the next chapter. This is the entire structure of centrosome. Just now you studied that, and these are the centriole, the central part, that is the centriole. These are the microtubules arranged in the way like triplet form in the peripheral way, right? So this is the structure of centrosome. So the centrioles form the basal body of cilia or flagella. So centrioles form the basal body of cilia or flagella, and that is what called as hub. As I was telling you that the central part, this is the structure seen in your diagram in your textbook, that is the central part, central core, that is called as central hub, which shows the peripheral triplet forms of microtubules which are made up of tubulin. So these are the microtubules in triplet form, and this is the central core or central part, that is called as hub. So this is the structure of centrosome. Now, microbodies. There are few microbodies present in eukaryotic cells. Two examples of this are peroxisomes and glycosomes. What are these? These are often called as, commonly called as microbodies. Peroxisomes and glycosomes, they are spherical, membrane-bound, minute organelles found in plant cells. Especially, these are present in plant cells, especially glycosomes, peroxisomes. These microbodies are spherical, membrane-bound, minute organelles found in plant cells. Peroxisomes' role is what? They are involved in catabolism of long chain fatty acids, synthesis of phospholipids, and photorespiration. So just remember the function of these small membrane bound structures, spherical structures, which are very minute and present generally in plant cells. So peroxisomes, what is the role? That is catabolism of long chain fatty acids, synthesis of phospholipids. These phospholipids are formed, synthesized, produced with the help of these peroxisomes, and photorespiration. So that is the role of peroxisomes. They both, how they look, they are present in the eukaryotic cell and together called as microbodies. Then peroxisome role is what? Generally involved in the protection of cell from toxic effect of hydrogen peroxide. Hydrogen peroxide is considered to be harmful, toxic, and so from protecting the cell, it is involved in the protection of cells from toxic effects of hydrogen peroxide. Hydrogen peroxide, wherever it is produced, it is considered to be toxic in nature. So the cell is saved, or that is protected from the effect of, that is the toxic effect of hydrogen peroxide. So these peroxisomes are playing important role in what you can say, protection. These are just examples where glycosomes generally found in fat-rich germinating seeds like castor, and these contain the enzymes of glycoxylate cycle, which convert stored lipids to carbohydrates. So these are the castor seeds where these glycosomes are present. So this is what about the different organelles present in the prokaryotic as well as eukaryotic cell. Now we will be discussing the difference between prokaryotes and eukaryotes. Where the prokaryotes, which type of organisms are seen in prokaryotes? Monera. Most of the bacteria, they come under prokaryotes, and all other organisms, remaining all other organisms, they come under eukaryotes. When we consider the size, prokaryotes are very small, 1 to 5 micrometer, whereas eukaryotes are much larger in size compared to prokaryotes, 10 to 100 micrometer. Then complexity wise, prokaryotes are relatively simple, whereas eukaryotes, they are complex in nature. When you study cell wall, when we compare cell wall, usually it prokaryotic cell wall which contains peptidoglycans, whereas eukaryotes sometimes present, sometimes absent, but it lacks peptidoglycans. You know, where in case of animal cell, cell wall is absent, in case of plant cell, it is present. So that's why it is said sometimes it is present, but it lacks peptidoglycans. Then plasma membrane is always present in both prokaryotes as well as eukaryotes. Internal membranes, in case of prokaryotes, may contain infoldings of the plasma membrane but usually lack internal membranes, whereas complex system of internal membranes divides cell into specialized compartments. And what are these all compartments? These are all different organelles, as we discussed just now. So they why it is called compartments? Because that is giving a specific boundary, right? So that's why they are called as compartments. Membrane-bound organelles are absent in prokaryotes, whereas all these are membrane-bound organelles resulting into compartment-like structures. They are present in eukaryotes. Ribosomes in prokaryotes are smaller and free in the cytoplasm, whereas in eukaryotes they are larger and may be found in bound to ER, that is endoplasmic reticulum. You know that. So in case of prokaryotes, 70S ribosomes are present. In case of eukaryotes, 80S type of ribosomes are present. Then cytoskeleton is absent in prokaryotes, whereas it is present in eukaryotes. Flagella, they are solid and made up of flagellae and they rotate, whereas they show microtubules bend in case of eukaryotes. Then genetic material is in the form of single naked circular DNA molecule in case of prokaryotes, whereas in eukaryotes, many linear chromosomes, each made up of one DNA molecule joined with protein. So such type of structure, linear form of structure is present in sometimes double nature, sometimes single nature. That type of structure is present in eukaryotes, whereas in case of prokaryotes, this genetic material is in the form of single circular naked DNA molecule. Location of genetic material is in the cytoplasm, that is called as nucleoid, whereas this is bound or it is present inside the membrane bound structure called as nucleus in case of eukaryotes. Now the last one more point which we will discuss, which you have already studied in your school, that is the plant cell and animal cell. Few basic differences we will discuss between these two. That is, a plant cell is usually larger in size, whereas comparatively animal cell is smaller in size. These are the structure which we have learned already. In case of plant cell, it is enclosed by a rigid cellulose cell wall in addition to plasma membrane. Means plant cells show the presence of rigid cell wall generally made up of cellulose, whereas animal cells lack this cell wall, but that is the structure, the outermost layer is plasma membrane. It is enclosed by thin flexible plasma membrane. Generally, plant cells do not change its shape, but animal cells can often change its shape. Plastids are present in plant cells, which are meant for different purposes. For example, chloroplast for photosynthesis. Such type of plastids are absent in animal cells. Then a mature plant cell contains a large central vacuole. That means vacuole is larger, and though few in number, its size is large in case of plant cells, whereas in case of animal cells, it possesses many but very small size vacuoles. So these are few basic differences between the plant cell and animal cell. Then these are few more points which can be considered as the differences between the plant and animal cell. That is, nucleolus lies on one side of the peripheral cytoplasm in plant cell, whereas generally it lies at the center in animal cell. Then centrioles are usually absent in case of plant cell except motile cells of lower plants, whereas they are present in animal cells. This is the specific feature that centrioles are present in animal cells, but in case of plant cells, they are generally absent except few lower plants. Then lysosomes, their structure and the number is less. Then glycosomes may be present in case of plant cells, but they are absent in animal cells. Just now we were talking. Then in case of uh, one more thing, plant cells, there are few more differences, that is spindle formed structures during the cell division. The astral and anastral type of structures are seen here, whereas in case of animal cells, this point you will not understand right now, so I will not discuss, because that during the cell division you will understand what exactly it is. Cytokinesis occurs by cell plate method, whereas it occurs by constriction of furrowing. That point also you will be understanding during the cell division when we will be discussing. So just few points you remember which are considered to be the differences. There are many more differences. Now here I have listed 15, but at least 8 differences you should learn. Common points between plant and animal cells, that means the organelles. In case of plant cell and in case of animal cell, here you can see uncommon part and here you can see the common part. Purposely I have shown this part, like comparison of the plant and animal cell. When we compare the organelles in plant and animal cell, you can see these are specifically present only in plant cells and these organelles are present only in the animal cells. But these are the organelles which are present in both plant and animal cells. That is the common. These are common organelles like plasma membrane, endoplasmic reticulum, mitochondria, nucleus, and Golgi apparatus or Golgi complex. These are present both in plant and animal cells. Whereas cell wall, chloroplast, and large vacuoles present only in plant cells, whereas centrioles, centrosomes, small vacuoles, cilia or flagella, they are present only in animal cells. So this is what comparison between the plant and animal cell. I hope you have understood, and whatever that is the end of the chapter. We have already discussed all the parts, all the organelles, structure of prokaryotic cell, structure of eukaryotic cell, then different organelles present in prokaryotic cell, different organelles present in eukaryotic cell. So today what we discussed, I will revise within a minute. That is nucleus, its structure and functions. Then cytoskeleton, all different parts or the different categories present in the cytoskeleton, like microfilaments, microtubules, intermediate filaments. Then we studied the structure as well as functions of cilia and flagella, that is the ultrastructure, that is. And then centrioles and centrosomes. Then we discussed the difference between the prokaryotic cell and eukaryotic cell, and then we discussed the difference between the plant cell and animal cell. And right now, if you have any doubt, you can note it down there. In the next lecture, next session, we will be discussing, solving all your doubts.